In my presentation, I would like to share with you the things that are close to my heart as a pediatrician. I have been in clinical practice for almost 4 decades, and I am often present at the birth of a child. These are joyful moments, but sometimes also very stressful, because it is about the survival of the child. We resuscitate the child and so on. Human reproduction and birth is absolutely central to the survival of the human race. Moreover, reproduction, including human reproduction, must have been a central process in the evolution. One of the key principles of evolution is that offspring with more favorable traits will survive and have more offspring than individuals with less favorable traits. I will quote from Charles Darwin on the origins of species. Now, if any slight innate change of habit or of structure benefited an individual, it would have the best chance of surviving and leaving offspring, unquote. The process of reproduction is taken for granted in the evolution, but the question is it a given? Is really the obvious thing? I could make a subtitle of this presentation, something like a preliminary case study in the high improbability of human common ancestry. And we will speak, we will look shortly at 4 steps in human reproduction. We will exclude the embryogenesis, which is very complicated. So I would like to speak only about fertilization, human embryo implantation, precise mix of hormones regulating pregnancy, the birthing process, especially about special rotations, fetus emergence, and birth assistance. And then we would like to look at this process from some perspectives. We would like to look at it from neo-Darwinian perspective, then from engineering perspective or systems biology, this very popular term today. And then I would like to deal with the argument from flawed design. So let's start with the first stage. This is fertilization. Between 100 to 300 million sperm up at starting line. And the first sperm to penetrate the egg membrane wins. The sperm are well equipped for the race. You can see the long tail they have to propel them forward. Along the way, they must overcome some significant obstacles, such as acidic conditions of the female reproductive tract, physical barrier of the cervix, and the attack of defensive army of white blood cells. Of the millions sperm at the starting line, only a few hundred will reach the egg. And reaching the egg isn't the end of the battle. The egg is surrounded, as you see, with a thick outer layer called zona pellucida. Fortunately, sperm are equipped with a mechanism to get past this barrier. The head of sperm is covered with a structure called acrosome. When the acrosome touches the zona pellucida, it releases just the right digestive enzymes to tunnel through the thick layer. The sperm membrane then fuses with the egg membrane. However, there are still many other sperm that would like to enter the egg. If this were to happen, it would create a genetic chaos and would be fatal to the embryo. Polyspermy block prevents this catastrophe. First, the cell membrane of the egg is depolarized, and later the properties of the zona pellucida change, which stiffens and loses its ability to attach to other sperms. Many of the details of this process of the polyspermy block at the molecular level are still unknown. How the sperm find the zona pellucida is also a mystery. But one thing is clear, and I quote from a review: the entire fertilization process is extremely delicate and precise, and there is no doubt that multiple layers of safety mechanism exist to ensure the achievement of normal fertilization. These processes are common to all mammals, with only with minor variations. So we could speculate and say to ourselves, there was a plenty time, some millions, 100 millions of years to evolve such a precise mechanism. But let's go to a second stage. For my further talk, I would like to explain a few terms to you. The fertilized egg becomes a multicellular formation called blastocyst. The inner cell mass of blastocyst is called embryoblast, which will develop into embryo. The outer cell layer of the blastocyst is called trophoblast. This develops into much of placenta. The innermost layer, you can see on the endometrium, is the most important player in the female reproductive system. The myometrium is the middle and the thickest layer of the uterine wall. It consists mainly of smooth muscles. As we said, the next important step in human reproduction is the implantation or the nidation of the developing embryo. Embryo implantation is a multi-stage process. It begins with the apposition of the embryo to the endometrium. Next, the embryonic cells and the endometrial cells of the uterus establish close molecular contacts. This is called adhesion. Finally, the trophoblast cells proliferate intensively and allow the embryo to invade the endometrium. I would like to stress that this is a very controlled process of invasion, unlike, for example, in cancer. This is also an invasion, but very uncontrolled process. And while invading the embryo, cells continue to multiply. These cells are responsible for the formation of the so-called placenta and produce the main pregnancy hormones. The developing embryo and the placenta is a so-called semi-allograft. It means it is immunologically not identical to maternal tissue. Normally, such tissue would provoke an immunological response and exclusion. However, the invading embryo is not rejected by maternal immune system. This is called immunological paradox of pregnancy, and it remains a central question in reproductive immunology. Remarkably, immune cells, so-called natural killer cells, can kill intracellular bacteria of the trophoblast without killing the embryo, without compromising its viability. Successful implantation requires another thing. It is precise synchronization between blastocyst development and endometrial maturation. Indeed, there is a crosstalk between the embryo and endometrium. In this communication, the endometrium acts as biosensor. It is able to respond favorably to competent embryos, but less favorably to incompetent, poorly viable embryos destined to fail. Some of the general features of implantation are shared by all placental mammals. However, there are some unique features of implantations in humans. For example, In humans, the preparation of the endometrium for pregnancy is initiated in the second half of the menstrual cycle, even in the absence of pregnancy. It is unlike in other species. What is much more important is the type of implantation. In contrast to superficial implantation in most primates, the human blastocyst is pulled into the endometrium and so-called deep placentation occur, meaning that the entire embryo is embedded into the endometrial stroma. You can see it on the picture above, while on the other pictures are some monkeys and rodents and marsupials. It is quite another history. So there is a distinct difference in this kind of implantation. The route and depth of trophoblast invasion is also unique. In lower primates, the trophoblast invades via endovascular route. It is this on the right. This process is shown on the right part of this picture. In humans, there is additional invasion via so-called interstitial route, which extends very deeply through inner third of the myometrium. This is shared also with some chimps, great apes, gorilla, and chimpanzee, but I don't know if this is identical process. It is some type of deep invasion, but I couldn't find if this is really identical. This deep invasion of the trophoblast is necessary for adequate blood supply to the fetus and required for its proper growth. Many changes had to occur at the genetic, cellular, and anatomical levels to make this process possible. Thus, we summarize, the implantation is a complex process that depends on the coordination of controlled invasion, placental formation, induction of immune tolerance, mutual biosensing between the fetus and the endometrium, and some of these processes are specific to humans and must have emerged only very recently. The third point are the hormones, precise mix of the hormones that regulates pregnancy. Pregnancy requires a chemical and morphological changes. These steps are triggered by a series of chemical messengers known collectively as hormones. They control and synchronize the production of an egg, its fertilization, development of the embryo, and the final delivery of the child from the mother womb. The major hormones that regulate the pregnancy are chorionic gonadotropin, human chorionic gonadotropin, progesterone, estrogen, prolactin, relaxin, oxytocin. However, there are many other hormones, as you can see on the right part of this slide, but we will not touch them. After implantation is initiated, this process, the basis of this process, is the switch from hormonal control of the menstrual cycle. You can see it on the upper part of this picture, into the pregnancy. After implantation is initiated, the embryo actively secretes HCG, so human chorionic gonadotropin, which can be detected in maternal serum as early as the 8th day after ovulation. The primary role of the HCG is to prolong the biosynthetic activity of corpus luteum, allowing continued progesterone production and maintenance of pregnancy. Again, although many of the processes involved in the hormonal regulation of the pregnancy are shared, with other mammals, there are more specific areas that are unique to humans. Humans have 6 transcriptionally active CGB genes encoding the active beta subunit of ACG. This is the highest number among primates, including the great apes. In humans, progesterone secretion is maintained until the onset of the labor, in contrast to many species in which parturition is initiated by the decrease of this hormone, of circulating progesterone. So it is thought that the functional progesterone withdrawal occurs at the receptor level, which appears to be a process very specific to humans. There is evidence that specific progesterone receptors have emerged rapidly in human lineage, making animal models of progesterone signaling inadequate for human biology. I have also found some differences, specific differences in genes for relaxin. There are 3 genes versus 2 genes in great apes, and GnRH hormone, which is also involved in human reproduction. But this is not all. The most interesting is the birthing process. This part of reproduction seems to be the most specific to humans. Human birth differs from non-human primate birth in 3 fundamental ways. First, there is the specific rotation of the fetus during birth. In monkey, birth canal is both inlet and outlet are largest in sagittal dimension. You can see it in this line, in this column of Penn. It's always the sagittal plane. In humans, the long axes of inlet and outlet are perpendicular to each other. So it starts with frontal plane, and then the outlet is a sagittal plane. The extremely close correspondence between the fetal head and maternal pelvic dimensions requires that the neonatal head and body undergoes a series of rotation during birth. On the left is the rotation of the head in humans. On the right of this left part, you can see the rotation from the entering the pelvis, and then is at the outlet. And in chimpanzee, there is no rotation of the head during the birth. But it is not only the head that rotates, but also the shoulders, which are broad and stiff compared to those of the great apes. The manner of fetal rotations during birth is specific to humans. Once more, you can see the entrance of the head during the birth, this initial stage of the birth in chimps and humans. And on the right part of this slide, there are rotation of first of the head and then the shoulders. This is one of the last but one picture. There is a difference how the fetus emerges the birth canal. In monkeys, the newborn baby is born facing the same direction as the mother. This allows the mother to easily reach down and guide the baby as it emerges from the birth canal. The mother can wipe the mucus from the baby mouth and nose to help it breathe. Other animals observe mostly the birth process from a distance and do not assist the mother or infant. The human fetus emerges from the birth canal in the opposite direction from the mother. It is difficult for the mother to reach down and clear the airway to remove for example, the umbilical cord around the neck, and to help the infant anyway. If a human mother tries to assist the delivery by guiding the infants out of the birth canal, there is a very high risk damaging the fetus. Spinal cord, brachial nerve, and muscles can be damaged during this procedure. So it seems to be that the response to this challenge is the assistance during birth. Virtually all women in cultural groups seek assistance in childbirth from relatives, midwives, or obstetricians. It is argued that midwifery is the oldest profession, not the other one, but midwifery is the oldest profession. In addition, human newborns are typically quite artificial, helpless at birth compared to the infants and of other species. Both assisted childbirth and the care of artificial infants are best performed by bipeds. And so it seems that bipedalism must have been associated with the emergence of human birthing mode. But bipedalism is associated with anatomical changes in the pelvis. It has to be narrower to make walking and especially endurance running energetically efficient. And again, narrowing of the pelvis is associated with the change in birth canal, as we have already discussed. So all these changes are interconnected. And there is a lot of questions around how and why it came about. In 1960, Washburn coined the term obstetrical dilemma to describe the functional and evolutionary conflict between the large birth canal and small pelvis for bipedal locomotion. He suggested that the relative immaturity of human newborns was an evolutionary solution to this dilemma. Some scientists, however, have challenged this obstetrical dilemma. Nevertheless, one of the arguments for its existence is the pronounced sexual dimorphism of human pelvis anatomy. This means the difference between male and female pelvis, as you can see on the right part of this slide. On average, the female pelvis is relatively wider than the male pelvis. As I said, an important constraint in human birth process is the size of the head. This is related to the growth and ultimate size of the human brain. At birth, newborns have no more than 30% of the adult brain mass compared to 40 to 65% in the great apes. In humans, the fetal pattern of the brain growth continues a year or later after birth. Hence, the evolutionary hypothesis of immaturity of the brain at birth. It is true that human birth must be precisely timed to the time the head can still pass through the birth canal. Yes, I will affirm this claim. However, to call this brain immature is totally unsubstantiated misnomer based on evolutionary narratives. Although the human newborn is profoundly artificial, helpless, he or she is not immature at the birth. The term newborn is not immature at the birth. In contrast, he is perfectly mature in all aspects of the functioning of all systems. I see it as pediatrician every day. Let's look on these facts from three perspectives. The first one is neo-Darwinian explanation of the human reproductive process. There are only speculations about the birth canal in Australopithecus. You can see the remains of loosey pelvis. So extrapolate from this how the birth canal was. It is a little bit more speculation. And there are no complete fossil pelvis remains of the known adult female Homo erectus. There is no evidence in fossil record for gradual changes in birthing process. Evolutionary narratives of human reproduction appeal to the selective pressure of natural selection. This is a claim of evolutionary science. But natural selection do not have the power to create novelty per se, such as new genes, new regulation, etc. We can summarize some new aspects of novel aspects of human reproduction. So we have size of the fetus and brain growth, size and shape of the pelvic canal, bipedalism, timing of the birth, regulation, deplacentation, which is needed to fulfill the metabolic capacity of the fetus, and some new genes we have found for only 4 of them. And suppose we will assume that there was only one mutation for each of these new features, with exclusion of new gene hormones, we can suppose very generously 4 mutations. What is the waiting time for them? According to the model proposed by Sander et al., it would take 18 billions of years to fix mutations in a population about 10,000 of hominins. I am not expert on the mathematical models that simulate the waiting time for such events, but I am afraid that the time frame for such event is outside of any realistic cosmic time interval. The problem of reproduction is that this mutation cannot wait for each other. They must have occur at the same time or near at the same time. If not, there is a risk of infertility and the end of the evolutionary process. Please understand, the end of reproduction, the infertility, is the stopper of the evolution. We have really underestimated this process of changes that are important for bipedalism. There is a list of changes, 26 items that are needed for the endurance running of modern Homo, as was published by Bramble in Nature in 2004. So not one mutation, at least 26 or much more mutations in this only one item of bipedalism. I cannot speculate how many mutations there must be in pelvis anatomy, in other processes that are needed for reproduction. Let's look at the human reproductive system from another perspective. It is engineering perspective, or so-called systems biology, which is very popular nowadays. The human reproductive system is a typical coherent interdependent system. For such a system to function, many parts must come together at the same time. To simplify, let us visualize this system as a cogwheel with 4 wheels. The teeth fit together precisely and wheel turns smoothly. Several statements can be made about such systems. The greater the number of coherent interdependent parts required, the more work it takes to design, assemble, and fine-tune them into a functioning system. The second, the greater the number of coherent interdependent parts, the more work it takes to change or evolve one working system into another working system. Look now at the possibilities how this could change. So we have system 1, system A on the left side and system B on the right side. In all of the system is fertilization, implantation, hormones and birth, fertilization, implantation, hormone and birth. Let's suppose this is an ancestor system A, such monkey-like, and this is a human system. And we will try to make evolution of this system by small changes in individual subsystem that would then evolve into system B. These basic engineering principles can be applied to the emergence of the reproductive system in modern humans. Using the inference of the best explanation, the intricate complexity of human reproduction as a coherent interdependent system is better explained by Occamrason, better explained by design than by unguided processes. As I said, there are small but very significant differences between the reproductive systems of primates and humans. Switching from one system to another by making these small changes in individual subsystem can very easily disrupt the functioning of the whole system. And this will lead to the infertility zone on IVF clinic, if you want. And this is problem of common ancestry. Moving from this one system to another, to these changes, is not the solution. So the creation of the new functional system with variation in individual parts is very difficult to explain without a design concept. Wishful thinking or these evolutionary narratives and even claiming miracles in some secular articles are not the solution of this hard problem. I would like to explain why design is a better explanation for this similarity. How can we explain the similarity in human reproduction without common ancestry? The similarity across the animal kingdom is reality. It is no doubt about it. But at the same time, if I try to show you in the human reproduction, there are differences that seems to be insurmountable, but evolutionary processes. If I demonstrated you on these 9 mutations example. And the answer for this could be common design. What is common design? It is a very central idea which explains very much of this a big part of this problem. The intelligent cause, such as Creator, God, can reuse and move the same modules, such as genes, regulations, processes, and some concepts, the same modules to other systems without the need of direct material or physical connection. This is very important, without the need of direct material and physical connection. One of the main cases for common design are taxonomically restricted genes, so-called orphan genes. These are genes that are unique for defined group of organisms in lower taxa, such as genus, family, species, acting as determinants of the lineage-specific biological properties. There are no evolutionary homologs for this. They are really evolutionary parents. They have no evolutionary parents. They are orphans indeed. In humans, there is estimation of up to the 1,000 to 1,500 taxonomically restricted genes. How many of them are involved in human reproduction? We don't know, but it could be a great deal of them. So this makes the neo-Darwinian explanation even much more problematic and complicated. Let's remember the basic idea with which we began. For evolution to occur, the reproduction of the offspring must be assured. Any setback in reproduction stops evolution. If we look more closely at the problem of human reproduction, it seems that this is the process. This process is the real bottleneck in human evolution. It is an evolution stopper. To my knowledge, this problem seems to be greatly underestimated in the evolutionary science. There are some arguments for human uniqueness. This is from my book In Vortex of Evolution, one of the table, and I will not deal with these issues, but I would like to add to this table there is also uniqueness of human reproductive process. This is only this chromosome Y in human. In chimpanzees we dealt yesterday, so you can see how much the difference is. We will look at the human reproduction system with the arguments from flawed suboptimal design. Abstracted labor is important factor in maternal and neonatal mortality and morbidity in non-medicalized settings in the history, in the past, and even today in some region. And this may be perceived as flawed or poor design. Design of a coherent interdependent system includes trade-offs, some compromise, and choosing some optimal features to achieve overall functionality. This is what every engineer knows. You have to make some trade-offs, designing some system. Even when there is a trade-off in human reproduction system, the system as a whole works optimally. The humanity reproduces. And the argument from bad design to bad designer to no designer, and therefore unguided evolutionary process is a totally flawed, fundamentally flawed extrapolation. But something happened to human reproduction. We have a question. Why is the childbirth so extremely painful process for the mother, as you can see on this slide? Videotape studies of great apes during birth do not vocalize much in pain and do not grimace much in pain unless there is some complication. Why is the case in the human reproductive system or human reproduction? There is an option, the degradation of the original design. We can read in Genesis 3:16. To woman he said, I will make your pains in childbearing very severe. With painful labor you will give the birth. Every time I leave the delivery room, I am remembered of two things. First, something extraordinary has happened. A new human being has been born. And the word is beautiful. Second, but the pain of the childbirth, which I often witness, tells me another thing. Something is wrong with this world. I would like to end my presentation with a look at our two lovely granddaughters who successfully went to the reproductive system a year ago and 2.5 years ago. And although I would like to show you the covers of the book, I have of the books I have published in the past year, both in the field of pediatrics, at the intersection of the Christianity and medicine. This is a book, is there any sick among you? It is it's based on my experience with sickness, alternative medicine, respect for life. These 2 volumes between Bible and medicine are the proceedings of our Christian Medical Fellowship conferences. And the Vortex in Evolution, I will tell you something more about it. And these books from the field of biblical teaching, I taught our youth in our congregation, in our local church, biblical truths some 15 years ago. There are 24 lesson session for them, and then I transformed it into the book, these books, and it was published with reprints. So this was a hobby for me. Let me add a very short testimony from my life. I grew in a Christian family, and at the age of 13, I committed my life to Jesus Christ. I have been always interested in the natural sciences. While studying at the university, I immersed myself in evolutionary science and evolutionary theory so deeply that I almost lost my Christian faith. I had very good contacts in then German Democratic Republic, where I participated in meetings for students, and this was very helpful for me to search for the answer. You can see the cover of the book by Professor Weider-Smith. I read then in German, Die Naturwissenschaften kennen keine Evolution, the natural science no evolution. And it was a book that probably helped me very, very much. Then years passed, and I devoted myself to pediatrics and to my clinical practice and my work as a consultant pediatrician. I do this. I work in this position for 28 years, teaching our new doctors. And about 10 years ago, I came across a book by Jonathan Wells on junk DNA. And I was amazed how far the scientific knowledge and the arguments for creation had moved from the time I started with this first book. Gradually, I began to study it, and I felt that I owed a certain depth from my youth that I should write something on this subject. For strategic reason, the ID approach appealed most to me. I still didn't have enough time to write, but then came the pandemic, COVID pandemic, when paradoxically I had more time, and I also needed to disengage from the stress and fear from the disease. It was terrific at the beginning. So it was a way to disengage from this stress. And so the book In Vortex of Evolution was born. I tried to incorporate the latest findings and newest knowledge in this book. You can see the table of contents. There are 5 chapters on origin on life, and then on Darwin pillars, on natural selection and common ancestry. I dealt with devolution, explosion in fossil records. So this is about this Cambrian explosion. Then I deal with junk DNA, epigenetics, common ancestry. About man, there are two chapters. Non-adaptive order in biology, Darwin on design. There are some philosophical thoughts also. Worldview influences on science. There are two appendix, science of and the God hypothesis, and dealing also with God of the gap, contradiction, which is very important, contradictions of theistic evolution. I wrote it also as a polemic with one of the Catholic theologian in our country that claims such very strange beliefs that matter is intelligent plasticine and so on. And then is a word of conclusion, my testimony about faith in God of Bible as a source of formation. It is 380 pages with 45 diagrams and figures. I wrote it specifically for Czech Republic because many English sources will not be translated, so I tried to make a research in this area. I have made some trials to translate it into Polish because it is my mother language. The Polish language is my mother language, and with deeper, but it can be read, but I think it is not a good translation. But it can be. I can share with some of them.